Well, good afternoon. Thanks for joining us again for part of the Data-Driven Ag series, in part sponsored by Digital Ag, and part the Institute for Digital Forestry and the Plant Sciences Institute. So today we have Dr. Jian Jin. I'll introduce him in a moment, but this is the full series coming up. So we hope that schedule and interest, if your schedule allows and your interests are there, we hope to see you back. And just a reminder that they're only in person, but we are recording them. So if you happen to miss one and you're interested, catch us on the College of Ag YouTube channel. So without more about that. So today we have Dr. Jian Jin. He is an associate professor in agricultural and biological engineering. Previously had some time in industry with them named Pioneer. He is an innovator extraordinaire, also an entrepreneur. You'll sort of see that in the topic that he's presenting today. I think you will see applications of this handheld device across many plants, and maybe in the future animals. Who knows? The opportunities are endless. So I know personally of some of this work, having been on a couple of his graduate student committees, and I found it just to be very impressive in a novel way to get insights about the status of plants. So without further ado, Jian Jin, thanks very much for presenting. Looking forward to this.
Thank you for the invitation. It's really an honor to be here and give a seminar again on campus. Thank you for coming. I see a lot of old friends, new friends, collaborators, badminton friends, pickleball friends. So here. So I know it's 12:30, and this is usually the time you are still digesting your lunch. So sometimes the digestion is drawing your blood from your brain, and I know it's a sleepy time for many of you. So in order to deal with that problem, I prepared more than 100 pictures, more than 10 videos. So imagine you are not in the seminar room, you're really in a theater. So it's my role really here to entertain you, not to pressure you after lunch. So just sit back and enjoy. I'm just going to show you a lot of the pictures and the videos, but hopefully at the end of the show, you can put all the contents together, and hopefully it can make some sense to you, and you can get some story to take back home and maybe keep you thinking, and maybe promote some future collaboration opportunities as well. So that is really my goal here. So very brief resume for myself. I got my PhD from Iowa State, Go Cyclone. Yes. And then I had a little over 5 years of industry experiences. Some of you may be collaborating also with this company, DuPont Pioneer. The company has been keeping changing names. The current name is Corteva, many of you know, one of the biggest seed companies in the nation. And in 2015, I joined Purdue as a faculty member. So as Dennis just introduced, I'm also an entrepreneur trying to commercialize some of our innovations, technologies onto the market, try to help more people to benefit from plant sensing technologies. So I'm not necessarily following this framework, but basically my rough plan today is to firstly talk about some Purdue phenotyping facilities. So we have, especially starting from about 9 years ago, we almost launched one automatic phenotyping facility almost every year, one facility every year. And we have consecutive 4 years, keeping deploying new facilities. And some of them are just around us behind the lily, some of them are in the field. So I know many of you are curious, and I happen to be a major designer or contributor to those facilities. So just want to spend maybe 3 minutes to quickly go through the facilities and show you why they were built, how we're using those facilities, just for your curiosity. But the major point today is new innovations, new technologies, how we moved from the large-scale phenotyping facilities to only serve the researchers and reach grant owners, but move our targeted customers crowd towards the low-income people. We have, 600 million farmers around the world. How do we benefit more people from all kinds of the locations? That is really our goal. So basically, how to put the hardware and software technologies into people's hands at any location. We did a lot of work for the innovation, for the development. So I'm going to introduce today's plan is to introduce 3 of our handheld leaf scanning technologies, hyperspectral, microscope, and RGB. So we will hear some stories about that. And then I'm going to play a lot of the interesting videos, hopefully interesting videos to you, showing you how those handheld devices are now optionally operated by robots instead of human. So we also developed ground-based robots and drone robots to automatically deploy these sensing technologies to the field, to the greenhouse, to the growth chamber. Just to give you an idea of what we can really do now. Open your mind and then think about the potential for the future. And then the number 4 and number 5, these things are really optional. I really don't think I have enough time today to cover everything because I really want to leave 5 to 10 minutes for the Q&A part, because that's the funniest part, the most interesting part every time for the seminar. So if you're okay with this plan, I will go ahead. So these are the pictures of the 4 so-called high throughput phenotyping facilities we deployed here at Purdue starting from 2016. So these 3 are kind of part of my lab. So this is actually Purdue's first automatic phenotyping facility. So we use the conveyor system to deliver the plants to the high throughput hyperspectral imaging tower, which is this black tower here. I'm going to play a video to show you how it works. So in this facility, basically, we move the targets to the sensor. But this is a common mode and in industry. I actually just finished the designing of such a very similar phenotyping facility for DuPont Pioneer for Corteva. And I believe that facility in Johnston, Iowa, currently it's still the nation's biggest indoor crop phenotyping facility. It's a $40 million investment. I actually came to Purdue right after I finished the imaging part of that industry system. So this is a very common system. The plants are like sleeping in their bedrooms. And then it's like every day they want to go to Disneyland from your hotel, right? So you wake up from your hotel, you walk through the conveyor system to the Disneyland. And what do you do in the Disneyland? You take rides, right? So we have a lot of the rides for the plants to go through. And every time one ride is one station, the plant can go through the hyperspectral station, fluorescence station, CT, X-ray station. So every time when we go through one station, we use a unique technology in each station to scan the plants for a lot of data. So the data collection is really the point of the Disneyland. And then when they go through the land, we collect data from a lot of the different plants. So from the data analysis, we can analyze how different these plants are between the genotypes, between the different disease, how those will change our crops, and how do we predict the yield impact from all those different, we call it G by E by T, kind of the interactive factors. So this is a very, very common mode. And there is one issue always linked with this kind of mode, delivering the plants to the imaging system, because the plants are not supposed to be moving every day, right? Usually in the field, they are just standing here for the whole life. So moving the plants is disturbance to the growth, and that is actually causing a lot of the differences. Sometimes minor, you can ignore, but sometimes, depending on your research goal, you're concerned about those movements and disturbance. So so that's the reason we developed. By the way, we had some money in our hand during that time. Not a quite big industry sponsor the grant. So we were able to build, just spending another $300,000 we were able to build another gantry system underneath the greenhouse ceiling. So the ceiling of the greenhouse. So that this yellow gantry is kind of playing the role of a mock drone system, if you can tell. And this drone is carrying a lot of the different sensors, again, hyperspectral, LiDAR, 3D, RGB, all kinds of different sensors, just like the drone flying above the field. And in the greenhouse ground, we grow the corn, soybean, and other plants just to mimic our field in summer. But the benefit is we can do all season phenotyping, and there is no pitch yaw angle problem of the drone. We don't need to deal with the geo-referencing. And we can scan the whole room again and again for every 3 minutes with a very high frequency and time series imaging is, I'm a big fan of time series imaging because that helps me to keep track of the change, the tiny little change over the minutes for the plants over the day. So that is, in my opinion, that is a very valuable asset. So if these are really successful, people got very interested in these kind of facilities and using them. So then we thought, okay, we also want to image some of the field crops. So we basically copied and scaled up this greenhouse gantry to the field. So many of you may have seen this if you are driving along the highway 52, just in front of our Indiana corn and soybean innovation center. You can see this gantry just behind the innovation center. So it's a 150 feet long gantry, just one-dimensional. This is a little different from our brother universities facilities. How many of you have seen the University of Arizona's imaging gantry? Some of you have seen that. That's a huge two-dimensional gantry. We had a one-dimensional gantry, of course, for the first reason is we have a limited budget. In total, everything combining together, I only had $300,000, including the gantry, the facilities, the camera, computer, everything. So we were only able to afford a one-dimensional gantry, but the benefit of having the one-dimensional gantry is we can build this shadowless operating facility because the gantry can be sitting on the north side of the field. And we measured the highest sun angle in West Lafayette and carefully designed the top facility so that even when the sun is at the highest position, which is usually June the 21st, right? During the noontime, 2 p.m., because we're using daylight saving time and we are 1 hour off for some reason. Not 12 p.m., 2 p.m., 1:52 p.m. actually. So even at that time, none of this facility on top is blocking the sun from shining directly on the crop. And this is actually avoiding a big issue of our brother university, University of Arizona's two-dimensional gantry, which is applying a lot of the heavy shadows on the crops, and that is very difficult to calibrate. So, and also all of you know AAPF, just want to make sure I do not overclaim my credit. This is a lot of our Purdue teamwork, Dr. April Carroll, Judy Hickman put a lot of the efforts into the design, and we have a lot of the outside vendors design. So my contribution to this is really around the designing of the hyperspectral imaging. And this facility is using our lab's software for hyperspectral image processing. But these are all the imaging facilities we deployed for my first 4 years at Purdue. Sorry for the flipping of this video, but if you are okay to turn your head 90 degrees, basically every time the door opens, one plant is pushed in, and internally, actually, the plant is rotated to face the side view camera with the best angle because we have both top view and side view, 2 hyperspectral cameras doing 1 scan inside. And once the scanning is done, at this time, it's actually on the real time. We do the image processing on the spot, on the real time. And for the processed results, we use high throughput internet cable to push to our cluster systems underneath the basement of mathematics department for further modeling and processing. And then that plant is rotated back and pushed out. And then the next plant in, the door is closed again. So this is the cycle. this system did solve one issue for the microclimate. I don't think I have time to talk about that one, but basically we also use the conveyor system to shuffle the plants to the different locations because some of you know in the greenhouse the plants are undergoing different heat and lighting conditions. So we want to remove that one by shuffling the plants around to kind of average out the uneven treatments. So this is a video of the field gantry. So when we operate, this is the one button. Made it very easy to operate. Just push one button and the gantry will be moving the weatherproofed hyperspectral camera to scan with the push broom camera doing the line scanning back and forth. And we get this kind of the imaging results, looking at all kind of the indices for the plants. But one very interesting thing here I want to share very quickly is how severe is the diurnal change of the plants, or how important we should know that the imaging time really matters. The imaging time really, really matters. So you can look at this from this chart. The x-axis is the time dimension. So when in this is deployed in 2019, 5 years ago. So we start the imaging from the 30th day after planting, and if you're familiar with corn, that is about V6, V7 stage for the corn plants. And every morning we just started the imaging right after sunrise. And the scan of this field takes about 3 minutes. So every hour we can scan for 20 times. Over the whole day, we keep scanning until the sun sets. And in summer, we have the very long daytime. So that gives us hundreds of images for the same field, just within one day. And I know many of you will ask me, why do you scan so many times? It's the same day, it's the same field. You are wasting your resources. But if you look at the results, we are just taking one index, the NDVI normalized difference vegetative index as one example, probably the most popular hyperspectral imaging result. If you take this one as one example, take a look at the change within just one day. And every day from morning to the noon to the afternoon, you see a very stable V shape. And this V shape's amplitude is very, very big. You see from 0.85 all the way to something around 0.7, almost 15% to 20%, 15% to 20% amplitude for the NDVI measurement differences. This cannot be ignored, because think about why we're doing drone remote sensing. Why do we fly the drone over the field? For example, Corteva fly the drone over the field to measure nitrogen usage efficiency. And for breeders, if you can find one genotype which is having just 2% higher nitrogen usage efficiency, this is huge for the company because we can produce much better seeds, much better yield, right? But if we're doing that among 20% variance, your noise is way too bigger than your signal. And that is not a good story. So this is not my focus for today, but I really, since we talked about the facilities, just want to share with you some of our discoveries. We published a paper on this kind of the diurnal analysis. We discussed with a lot of the plant scientists, especially from Lily Hall, to talk about why this is changing. Is that because of the sunlight, or because of the biological activities, or because of the physical gestures of the leaf? A lot of the interesting discussion and research. And for those of you who are interested in more details, you are so welcome to check our papers on the diurnal analysis. But all those researches cannot be done without the construction of these facilities. So my point here today is really just to show you one example why these facilities are important, because without this gantry, we can't reveal this kind of a very interesting V-shaped. And for the first time in the world, we quantitatively characterized this V shape of the diurnal for NDVI for the first time. So without the facility, we can't do this. So those are about the facilities, and I already used 70 minutes. And now let's come to the most important topic. Question?
So on the previous slide, I can see that you have a waving condition.
Yeah.
So the year types of the
yeah.
What is
yeah, a very brief answer to that is actually the change. The change in the imaging result is actually caused by a lot of the factors. It's not only the time, we call it diurnal change, that's just about the time, but wind speed, humidity, rainfall, temperature. Actually, everything is impacting our imaging result. That is why we put the mini weather station here. We actually correlated a lot of the multivariable weather conditions with our imaging result and determine which factor and how they are impacting our result. Again, for details, you can check our paper, but that's a great question. Thank you very much. So the major point for today. Yeah. Thank you for reading our papers. So, we are so proud at Purdue to have the facilities. Purdue is one of the leading universities in the whole nation, and we should all be proud of that. And thanks to leaders like Karen Plaut and Bernie Engel, so they really supported these constructions. So we are so lucky here at Purdue, we can have access to those facilities. But think about a lot of the other researchers. These facilities can only be afforded by big companies and big universities. You can think about just several: University of Nebraska, Lincoln, University of Arizona, Illinois, probably Iowa State. Otherwise, I don't think of too many universities who can be as lucky as us. So the question is, how do we already know hyperspectral imaging, for example, is the very advanced and useful technology, but how do we benefit more people? So the idea is looking at our phenotyping facility. It's basically about hardware and software, right? Can we squeeze the hardware and the software and implement them into a small handheld device like this? So we can put both hardware and the software of hyperspectral imaging technologies to people's hands. So we can use the technology at any location in the field and in the greenhouse. So the good news is we did it about 7 years ago in 2017. So in 2017, our lab, the ABE Plant Sensor Lab, we were very proud to release the first version of LeafSpec, which is a handheld hyperspectral single leaf scanner. So before we talk about this LeafSpec technology, another motion or another reason for us to miniaturize the technology from kind of the remote sensing or the big imaging tower into a handheld device. It's because a lot of the bothering noises as well. So for example, for those of you who are familiar with our imaging tower or the drone remote sensing, for example, if you fly the drone like this, you use the sun as the lighting source, right? But the sun is a very unstable lighting source. It's keeping changing the angle, the color, and the intensity. We enjoy watching the beautiful sunrise and the sunset because it's a beautiful red sphere at that time. But right now, don't look at the sun. It's very dangerous. It will hurt your eyes. So the sun is dramatically changing over every minute. And also, the clouds are moving. It's applying shadows to the ground. So that is why we need to calibrate. And we know there are a lot of the calibration methods. You put white references on the ground, you can do all those referencing. But if you trust me, I have been doing those referencing for many, many years. There is just no way for you to accurately calibrate all those lighting changes. You are suffering from all those lighting noises. So this is one of the issues. Another issue is take a look at if we take a remote image of the crop, what kind of the noises we are suffering from. It's basically a combination of all kinds of things in this image. Did you see that? We have the very bright pixels. And these pixels are actually like mirrors. These are the mirror reflection of the sun. You're actually imaging the sun through the leaves as mirrors. So you are doing sun phenotyping, not leaf phenotyping or plant phenotyping. And compared with these very bright pixels, you will have a lot of the shadowed pixels. So these are totally different pixels. And also we know we have plant scientists here. We know the top younger leaves and the lower older leaves. They are totally different. That difference is way much bigger than the genotype differences or the treatment differences we are trying to research. So again, noise is way much bigger than signal. But for most of the current or all of the current remote sensing work, including my own work, I have been flying drone in the company and here at Purdue. what we do is we simply average through all the pixels of one plot. So we call it plot level remote sensing. We just average everything together. And in statistics 101, the professor will tell you if one data set, if the internal variance is too big, you should not use the mean value as the representative of the whole data set. But we have been doing that for so many years. And this is bothering us, but it's so hard to change this because it's just so difficult because we don't have enough resolution from so far away to really distinguish between all those different objects and analyze also even the leaf angle, the leaf slope is a big factor of that imaging result as well. And at another time, we really like that resolution because a lot of the diseases show up as spots on the leaf, and you cannot see this with a drone or even with our hyperspectral imaging tower in our facility. This is very hard to see. Between healthy leaf, the potassium deficient leaf, and nitrogen deficient leaf, you see the yellow surrounding green, green surrounding yellow, that kind of the differences. This is actually very valuable information in phenotyping, but without enough resolution, you can't do that kind of analysis. So combining all those are just the reason why we feel, it's probably a good idea to do some imaging with another mode. How about we image in this mode? We capture the leaf and slide through the leaf. And then we get this kind of image. I don't know if you realize that if we do the imaging like this, is there any ambient lighting problem? Is there any leaf slope problem? Do we get the high resolution? And, and we can control the leaf, put the leaf really flat. There is no leaf slope issues. So we got so excited about 8 or 9 years ago when we thought about brainstorming about this kind of the imaging mode. We got so excited because looks like all the major issues which had been bothering us for so many years can be just resolved by this new imaging mode. So we got so excited, and we are number one, not for this year, hopefully next year, but we have been number one ag engineering department for so many years, and we are engineers. We can design and develop things, right? So why don't we put the hardware and software technologies into a handheld device and scan it like this? So after the scan, we get this beautiful hyperspectral image. This is real hyperspectral image. Each pixel has hundreds of different colors compared with only 3 colors in our human eyes. That is why this device can see way much more than human vision capabilities. Number one is hyperspectral. Number two, this is probably the unprecedented spatial resolution. You see the midrib, you see the secondary lens. And basically, by the way, this is actually the chlorophyll heat map generated as one of the results from the hyperspectral scanning. So you can see the base of the leaf is losing chlorophyll. The edge and the tip is losing chlorophyll. But there is kind of like a gradient from the left side to the right side. This is a lot of very valuable information for you to analyze, to compare between the different stress patterns, disease, nutrients, and the different growth stages. They should show up as a different image in this kind of heat map. How can we apply AI, deep learning, to analyze this kind of the pattern, to reveal what kind of the stress, more like even in a regression level, it accurately predict the nutrient status and things. So spatial resolution is second thing. So it's hyperspectral, very high spatial resolution. And combining hyperspectral and spatial resolution, this is a completely new data set to support a lot of the analysis of the plant health. And also, we removed a lot of the noises, as we just mentioned, so it's high quality. So these are the 3 highlights: high quality, real hyperspectral with hundreds of bands, and a very high spatial resolution. So for the first time, we were able to generate a leaf image like this in a high throughput mode. So I believe this is the reason why LeafSpec got a little popular nowadays, and we have many customers asking to purchase LeafSpec and scan leaves like this, so we can do totally different levels, micro levels of the analysis for the diagnosing of the leaves. And by the way, a few minor points. Since we are scanning and the scanning result is pushed, you probably saw the smartphone app. We are talking through the Bluetooth. So just a seconds after the scanning, we can see the water, drought stress, nitrogen, and other disease information on the phone. And since we already have the phone, why don't we geo-reference the result and timestamp the result and upload the package to our NSF-sponsored data infrastructure? Thanks to Dr. Carol Song's support, so we're collaborating with NSF project for the GIS system to show our results. So it's geo-referenced. And on the spot, you can see all the measurement results. And we have one term saying one scan, do it all, because this one image is so much information. We have the insight into the leaf with hundreds of wavelengths, and we have that spatial resolution. And now we have AI. We know how much ChatGPT can do. Imagine when ChatGPT is involved and trained to just deal with this kind of the LeafSpec image, hundreds of wavelengths for each pixel and tens of thousands of pixels on the leaf. And we know exactly which pixel is from which leaf. Imagine how the smart ChatGPT can tell you about what is going on with the leaf. So that is the future. That is the goal, why we developed this work. So we talked about LeafSpec, and by the way, we also have a lot of the different versions. The first version of LeafSpec is for corn and sorghum. It's because of my collaborators, my major collaborators at Purdue. But then many dicot researchers got interested. So we had a master's student who spent the whole master's program, developed the soybean, the dicot version. It's the same idea, hyperspectral single leaf scanner. And then we collaborated with industry companies like FMC who are studying wheat blast disease. So we developed a smaller version of LeafSpec, basically still keeping the very high resolution, even just for that very tiny, thin, fragile wheat or rice leaves like this. And we have a multispectral version, which is more compromising between hyperspectral resolution and a spatial resolution, just to meet different people's needs. So this is a family we have for the LeafSpec family. We have to provide the different options for different customers. And this is just one example of how we use that in the field. So every summer, we are so busy using multiple devices, scanning here, 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 and here. And after a couple of hours, we can view all the results from the map. And then, currently the digital data infrastructure system can provide us a lot of the viewing functions. You can draw the contour map, like the nitrogen map. We can predict very accurately for the whole field, which can be further transferred to the fertilizers, digitally controlled fertilizers. So we can save that huge waste of over-fertilized nitrogen with this kind of the technology. All the potentials for the beautiful future. So we have this. Dennis, maybe you can help to pass this along. So we actually 3D printed the chlorophyll content heat map for the high nitrogen soybean leaf and low nitrogen soybean leaf. So usually, how do you tell between the high nitrogen and low nitrogen? Because we know nitrogen causes chlorophyll, and it's basically green versus yellow, right? But instead of just checking generally how green it is or how yellow it is, we can actually do this kind of a fine level prediction of the chlorophyll contents. And then ignore this complicated formula, but in one sentence, this is my PhD student, Dr. Zhihang Song's work. So he realized that the biggest difference between the high nitrogen soybean leaf and the low nitrogen soybean leaf is not the average color, but the contrast between the secondary vein. Like you see, these are like the ridges, these are like the valleys. So how from the ridges and the valleys, they are contrasting each other. And he somehow invented this index. And with this index, the data tells the story. The data shows that this index works much better than the averaged color in telling the nitrogen. By better, it means we have a much higher correlation with the ground truth nitrogen measurements, or we can tell the difference between the high nitrogen treatments and the low nitrogen treatments many days earlier, around at least 5 to 7 days earlier, before the averaged color can show some statistically significant differences. So that is why another example of how the spatial resolution combined with spectral resolution we can do better. L is a number of bands, things like that. Sorry about that. But you can probably, we can talk for more details. And this is actually published paper. So read our papers. No offense, read our papers. So I really want to I'm going a little more slower than I want, but I really want to show this exciting image here. I hope you can get this image. Basically, the image is about combining spatial resolution and spectral resolution. And for those of you who had some experiences with hyperspectral imaging, you know, for each pixel, we have the wavelengths from 400 nanometers all the way to sometimes 2,500 nanometers. And with different wavelengths, we can calculate tons of different indices. Many of you have seen these indices. Usually, for example, the NDVI is what we talked about. And each of these indices are usually just kind of like a ratio, normalized ratio between two wavelengths. And you know, think about this. If we have 500 wavelengths for LeafSpec, for example, and then we can pick any of the two, randomly pick any of the two wavelengths and calculate a similar index like this, right? Just the ratio between two. How many such indices can we generate? 500 choose 2. I don't know the exact number, but should be dozens of thousands, right? So just for this type of indices, we can generate dozens of thousands of heat maps. Not to mention, we can complicate the formula and calculate a lot more different indices. So we have, for each index, for each of these index, we can generate a heat map, like the chlorophyll content heat map or the NDVI heat map. So we have at least dozens of thousands of different indices, heat maps we can generate. For each of them, it's a new heat map. And for each of the heat map, because of the spatial resolution, we can look at the edge, the middle, the tip, the base, the middle, and we can follow the band development direction and see along the growth from the younger part to the older part, how this is changing. I hope you're following me. If you're following me, there are so many options of the indices, spectral indices we can generate. And there are so many ways we can check on the spatial dimension. We can calculate the texture feature. Here, we can calculate just the yellow color here, and then combine everything together and use AI to make some judgments. Basically, my feeling is, I'm not sure if you follow me, but my message is we realize that combining the spatial resolution and the spectral resolution and the spatial resolution, this is the explosion of information. This is very hard for you to process, but there is a huge potential for you to develop new diagnosing tools. So for example, one of our commercialized software using LeafSpec image, it can easily generate over 20,000 spatial spectral features just for those simple images. And with 20,000 features, you can do another multivariable analysis, partial least squares, support vector machine, deep learning. And try that and compare that with just the average color spectrum modeling. If you have followed me, you probably see the potential of how much better we can do. This is a new year. I believe we should be able to do much better now in planet diagnosis. And this is one of the examples. We talked about tens of thousands of different spatial spectral features. But in this chart, I'm just listing a few of them. Probably we have 40 rows and maybe 50 columns. So maybe a couple of thousand different spatial spectral features. So each of the x-axis, each of the column here is basically a different index. So for each index, if you look at the performance of this index at any specific locations, spatial locations on the leaf. So for example, sample location, we can divide the mid-rib into many different sections and look at the first section, second section, third section. You know, all these are just like zip code of the pixel on the leaf. The leaf is like a map. Each pixel can have a zip code. So these are just like the zip codes. These are like the indices. So we can take a look for each index at each zip code location. We can just do a local prediction of nitrogen. And let's see the performance. We can just use the correlation between our prediction and the ground truth as the performance. If you do plan essay, you know what I'm talking about. And this heat map is basically about the performance of the nitrogen prediction. Sometimes they perform really well. Red, shining, colorful areas are good performance. The blue ones are bad, not so good performances. This is actually a lot of the information because we can tell some of the columns are really performing well. This means, for example, if this column is really one of the champion of performance, it's telling us the ratio between 610 nanometers and 700 nanometers is really highly correlated with nitrogen at almost all the leaf locations. So this is a really good index. And on this dimension, we see a very shining band here. And it looks like all these rows are something about mid-rib. This is something I didn't know before doing this analysis. I learned from this analysis that actually the mid-rib of the cone is much better compared with the other side, other portion of the leaf in prediction nitrogen. You know, usually before this, what I did by myself, I segment out the mid-rib because it's like noise. I don't like the mid-rib. It's a small thing in the middle. It's a different outlier in the middle of the leaf. I threw it away and I want to purify my image. But now I learned that mid-rib is actually the most important thing for nitrogen. And this actually makes sense because nitrogen is transferred through mid-rib. This is a pipe. This is a headquarter for nitrogen. And then we talk with plant scientists to understand this and to develop new algorithms to try to develop the better models. So really need to move forward. I will skip this one, but this really want to talk a little more about this one because this is again for nitrogen prediction. So usually before, you know, I'm not a plant scientist. I had my training background as an engineer. So I usually thought chlorophyll is like green pants painted uniformly on the surface of the leaves. That actually is not true. So if you look at the cross-section image of the cone leaf, you know, this is like the middle rib in the middle. And then we have the small secondary veins, one by one here. So we're looking at this, we realize that the chlorophyll is like green rings surrounding each of the pipes. A ring here, another ring, another ring, another ring here. So when we are losing chlorophyll, when we are losing nitrogen, it's not basically the green turning to yellow. No, it's actually the greenness is shrinking and thinning. The green ring is thinning themselves. When they're thinning, the gap between the two neighboring green rings will enlarge. And if you are following me, you probably can tell from the LeafSpec image, we can see that enlargement of the gaps. And this is one of the algorithms my PhD student, Dr. Jian Jin, he reviewed that by looking at the LeafSpec image on the surface. So we're doing the transmittance imaging on the leaf, but we can clearly see that when the leaf is losing nitrogen, the gap in the middle, actually this yellow band is actually corresponding to the middle hole of one secondary band. This is another neighboring secondary band. But maybe you can see this, there is another lighter yellow band in the middle. This lighter yellow band is actually corresponding to the middle gap in between these two bands. And when the plant is treated by low nitrogen, this gap will be enlarged and intensified. And so that Dr. Song was able to characterize this kind of a feature and introduce another new, this kind of micro level nitrogen index, combining spatial and spectral dimensions. And his data proved that, again, this is much better in correlation with compared with the average color, or he was able to detect the low nitrogen from high nitrogen many days earlier before the average color showed this. So I will skip this one. Just to give you the information, as Dennis just mentioned, I'm an entrepreneur, so I was encouraged by Purdue University. We have the tremendous environment here to promote commercialization. So we were encouraged by Purdue to commercialize LeafSpec. So you can buy LeafSpec if you can allow me to do some advertisement here. you can buy, contact me, and we were very proud to win the company were very proud to win the Davidson Prize in 2021, which was awarded to at most 3 greatest innovations in agriculture every year. We were very proud of that. And we have many issued patents around the LeafSpec already. So this is about LeafSpec, but very quickly, just maybe 1 minute for each of the other devices. So LeafSpec, as we said, we have the hyperspectral resolution, high spatial resolution, and high quality. But sometimes we need even higher spatial resolution. Some people are not that interested in the hyperspectral, but they are interested to see the micro tissue level, cell level stuff. So this is another innovation. So we call that the handheld microscope imager. Long story short, I'm not going to go through all the technical details, but basically the same way as you use LeafSpec, you can use this device to just attach it to the leaf, and only 10 seconds later, you can get the image like this. And you can see the stomata very, very clearly. And I know at Purdue we have faculty members, researchers, very interested in the closing and opening of the stomata for photosynthesis activity study. And there are just a lot of the values that if you can do the microscope imaging like this on the spot with a comparative high throughput pattern, just 10 seconds later, you can get this image. So by the way, our engineering contribution, I believe, is the auto focusing function. So we made the focusing so automatic and fast to enable this kind of fast imaging instead of you take the leaf back to your lab and clean the labs for many, many minutes. You know, your middle school laboratory life. But now we made it on the spot, in situ, on the real time, and you can take those very fine images. And a very good potential for this microscope device is that we clearly see the spores and the tubes of the fungus for a lot of the fungus diseases for corn, soybean, and other species. So this is one of the imaging results for the several of the major corn diseases, so that we can actually not only detect those spores and tubes on the leaf for those different diseases, but also we can tell apart between the different types of the diseases, and we can even tell the status of the development, of course, because by telling the density of the spores, by telling the tubes development, sometimes, of course, we see the spots as well. So we can do the very early detection of the diseases. And thanks for Dr. Talanko's support. Darcy is there. So we have a lot of the materials from her field and from our research field in Acre as well. And this year, you know, we had a very humid summer, and we got a lot of the images. So now we're developing this technology to try to provide the early warning of these signals by directly seeing the spores and the tubes. And by the way, I know I'm short of time, but I really want to share this personal opinion. Compared with nitrogen content prediction, I myself is even a little more excited about this spores and the tubes microscope detection, because this is something we already see with our eyes. This cannot just escape. And especially when you are having some entrepreneur way of thinking, you know, advanced technology is not what people need. Solid and working technology is what people need. And if you can see the spores and the tubes, you know the deep learning will not miss it. And you know no overtraining will be a concern along the way. So that is why I myself is kind of very excited about the progress on the microscope device. And then the third handheld device I will mention is the so-called Leaf Studio. So this is thanks to Dr. Fei's support. This is part of our digital forestry image library construction work. So in order to construct the image library of all the different tree leaves, we need to have a device to take very standard leaves. So how about using our phone? Because everybody has a phone in our pockets and everybody's taking images. And we do have a lot of the different apps helping us to take pictures. I know many of you tried some of the apps, probably you take a picture of the plant, it tells you the species of this plant. But in my opinion, no offense to all those, those are great apps. But as a professional imager, I'm not satisfied with imaging quality because of a lot of the different reasons. Because when you image this plant like this with your phone, you don't have control of the leaf slope, which is a big factor in the greenness and the color. The shadows and glares, again, the varying background. The background can be even water for other plants. And then it's a low resolution. You're not that close. So we are thinking about, can we develop a more professional imaging app for the phone so that we can take so-called passport headshot standard single leaf images. So that is why we developed a very low cost device like this. So you can put the leaf inside this one and you can see the background is a beautiful shining light box, which is shining on the leaf. So we can do the transmittance imaging again. And just put your phone here, there is a hole. You still use your phone to take the image of the leaf, but you have the control of the lighting. You put the leaf beautifully flat. So it's just like in our Walgreen passport headshot service. We prepared everything for you. You just move your head, face there, take a picture. And now the benefit is if all the images are as standard as these images, you have the beautiful resolution. We can even rotate all the images to have the same angle, and also all the spatial resolution, showing all those spots and the details. This provides a very rich data set for people to do further analysis, species classification, disease detection. And many people can use this. People can download. This is very easy to 3D print. You can download from our website and 3D print by yourself and produce your own set and contribute to our library. And then when all the researchers unite, we have a beautiful future for collaboration, right? So, so I really need to finish. But before I start any questions, I want to, if I can still spend 3 minutes. I want to play some videos for you. Those are the robots. Hopefully you will have some fun. I will not say much, but you see what these robots are doing. Basically, we have these robots to deploy. You know, when this is in AAPF, deployed in AAPF. Every time when the part is transferred by the conveyor here, we have one robot holding the stem, another robot, you know, holding the LeafSpec, doing the scanning fully automatically. Ignore these boys. They are not really doing anything. This is fully automatic. And we successfully demonstrated this for many times, especially this spring. But now, due to the construction, we moved that back to my lab. Hopefully, it will be back very soon. And then a soybean version as well. This is not. Yeah. So this is actually in that gantry greenhouse. So we just mentioned in that second facility. So we use machine vision every time to automatically, you know, detect the top mature trifoliate. And then the robotic arm will do the path planning and capture the leaf. This is our die-cut version of the LeafSpec doing hyperspectral scanning. And this is a corn version. This is a more complicated and slower version, just because corn, we need to slide through the leaf. But it worked pretty well. This is my PhD student, Archer Chen's master thesis in 2019. So he used the 3D and RGB machine vision to detect the color of the leaf, which is a sign of the maturity of the leaf. And then every time when we see the color, we know the leaf. And then we capture the leaf, scan the leaf using this self-developed robotic arm. The framework is carried by our rover vehicle and move along the roads. So every time you just stop at one location and scan a few leaves at each location. So this is a corn version. And then we have a very cool PhenoBee. This is our feature, the robot. We call it PhenoBee because it's like a bee flying to the different locations and landing at each location to examine leaves. So this is fully automated. Again, we can use the computer to program this drone robot to fly to multiple designated locations. For each location, the landing gear can be very accurately within 2 centimeters area inserted into the gaps and land. And once it's landed, same thing using machine vision. As you can see, we basically moved the greenhouse technology underneath the drone. So it uses machine vision to detect a leaf and then capture the leaf. So you can see for the soybean leaf, you don't need to slide through the leaf, but we capture the leaf and move the camera and the light inside the box to do the light scan. And once the scanning is done, you can sitting on your chair drinking juice and do your QC, check the spectrum. And then if everything's good, you don't need to do anything, but the drone will fly to the next location in the field. So this is a PhenoB. If PhenoB is still too slow for you, we have another option for you, which is really like a smaller bee. So for this one, this is not landing at all. It's just hovering above the field. And when it's lowering to a certain position, now you can imagine a lot of the deep learning boxes are looking for the top material leaves on canopy. And once we lock in a target, we lower down, and then we use a vacuum machine in the box to suck the leaf underneath the box. As you can tell, I'm a big fan of control the leaf before doing any imaging because I had enough with those noises. Every time I want to capture the leaf, and then I enjoy the high resolution and the low noise and the control of the lighting and other things. So I think I really want to stop here. There are a lot of other things we can share if you're interested to talk with me in the future, but I want to leave 5 minutes for questions. Dennis, you want to start?
Great. Okay.
Go ahead.
Yeah, that's really amazing.
Thank you very much. It's very nice.
You're developing. And LeafSpec, I see some features through your bee drone-based bee technology.
Yeah.
But I'm still wondering from the perspective of farm scale management. So LeafSpec is collecting vein level data set and also geo-referenced. But how we create a geo-referenced points if we want to think about farm level mapping or
farm level, yeah.
Precision.
Yeah. So I think data infrastructure is a big thing. So we do spot after spots, but then how to combine all those spots. It depends on the research goal. So for example, if you are talking about drought stress, a few samples is probably enough for the whole field because if one plant is dry, then probably your whole field is dry, except for those terrain flooding issue. But mostly it's the case. But for nitrogen, it's a higher variance. So for basically the different variance level of the field will require a different density of the sampling. So that is a question depending on each specific case. But so far we have been working on diseases and nitrogens. And on the commercialization side, we believe there is a very wide sweet spot talking about how many samples you need to take for a typical 2,000-acre field to get the basic idea of the disease and the nitrogen. This is one thing, because it's geo-referenced, and on the map, you can do that scaling up. Another thing is the data infrastructure can also help you to improve your efficiency and throughput. Usually when we talk about phenotyping throughput, we talk about how many samples you sample for every hour. But in my opinion, a better scale, a better metrics is how much information you collect every minute or every hour. Because for example, in the whole field, if you take a few random samples and realize your whole east side is okay, but your west side is showing some problems, you probably should stop wasting time on the east side and focus on the west side, right? So with the real-time imaging result, can you imagine that? Our later scanning can be helped by the earlier real-time reported information to change our scanning order in the field, to focus on those problematic areas. And this is a much higher efficiency of scanning. So hopefully that answered your question.
Yeah, sort of, but I'm more thinking from the angle of like if GPS coordinates are a meter off and we are scanning every vein in the leaf, we are still aggregating information within that 1 meter GPS coordinate uncertainty, right?
Sure.
So then how we can interpret that vein level nitrogen efficiency data set for like, making a precision management across the farm.
Didn't get the point. Can we
Yeah,
talk about this later? Maybe leave
Yeah,
some
yeah,
time
sure.
for other questions. But let's chat after this.
But it's a good we have a few questions. The first one, I think it's more related to sales question. How is your data is whether that is your taking let's say you have a geo data of land. You are taking data from a single land
Yes.
and then from a single leaf.
Yes.
Do you think that data is representative? Yeah. That's one thing. The other question, did you try to calibrate the hyperspectral image using the hyperspectral sensor? That's the other question. My last question is, how is your...
Hold on before I forget. Maybe responding to your first question, how many leaves you need to scan? Basically, that's the question, right? So actually, as many as possible. But that's all.
My question is like, so you have like, let's say you have some area. So you are picking one plant. Even from one plant, you are picking only a single leaf.
Mhm.
So so you collect data from that single leaf.
Mhm.
And you are going to say the chlorophyll content or something of this, the whole land is this much.
Mhm.
Do you think that data is representative?
No, you usually need replicates. Usually not just one. You need to decide on how many samples are needed to represent this whole field. And this is again dependent on the research. And this is a big question. Every time when we collect data, we need to decide on how many replicates you need for each area.
Relationship between the different points that you measure with the newspaper and phenotyping and the yield data, and spatial yield data, for example. We can do that analysis if you don't already have it, but I'm just asking if you already have it.
Right now here, I don't recall a specific paper talking about that, but we do have a lot of the geospatial data saved in our geodata hub, and we can talk about that.
Let me chime in a little bit to say that I think that the way how you're using these technologies, I like to scan all the leads to know what is happening. But the way how I see it is, this is in a very advanced setting, some kind of diagnostic. So for example, if you go to the doctor's office, they don't need to do the CT scan every patient. They just do some of the pre-screening analysis, see if some symptoms. Hey, I like to see whether you have that specific disease or not. Then you go to the MRI or CT and to diagnose that, hey, you have this kind of problem. So the way how I see it is that you're going to have other technology that will tell you, hey, there's some spot that looks suspicious. Then this device can go into the field, have a scan of those leaves, then that can give you some of deterministic determination. Hey, these tree might have some kind of species or not. So I really don't think. Sorry, I'm just
No, this is a good point.
direction, but this is not meant to do a lot of leaf scanning, but that gives you a final determination. Hey, these the plant has some kind of problem.
Yeah, and talking about go-to-market, right? If some technology can give farmers the earlier warning and help them to make the decision to call those metabolomics, nitrogen leaf sampling service, if that can save them some time, that will be of some value, right? So we are not only targeting at ultimate solution, but when the technology is developing, the future could be better. But there are still a lot of other practical things to consider. How many replicates? How good quality is enough? Things like that.
What about the calibration? For instance, we have a really hyperspectral insight. But you are going to want to look at the certain image your data with the handheld camera, right? You try to calibrate from this, hyperspectral.
Yeah, I did a lot during my industry work, but everything from my last job, I'm not allowed to say too much. Hopefully you understand.
Well, continuing conversation with Dr. Jin, I think. Thanks again for presenting,
Yeah.
and hope to see you back in a couple weeks.